is 16th of March, and it is our third lecture of the course Neurophysiology and Cognitive Science. This is our schedule, 8 lectures and 1 lesson for an exam. The next lecture will be in MIPT, and I'm going to bring some neuro devices, neural interfaces with myself, and I'll tell you more about it later. And that's why I decided to change the order of the lectures. So the next lecture after this will be on attention and meditation, but not on neuroanatomy, and we will have online lecture on neuroanatomy a week after. So this is the relevant schedule. And today the topic is brain research methods. So we will discuss different methods, MEG, EEG, fMRI, PET, TMS, and so on. As you remember, these are our communication channels. In Google Class, you will have all the next text. You'll have one more test after this lecture. I suppose after an hour, when the video of the current lecture will be created on YouTube. So I'll send you both links to the YouTube and to the test for this lecture. So let's move on to the lecture. As I said, our today's topic is the brain research methods, and there will be 4 parts. The first part is the short story. The second part is about methods to investigate the brain at the neuron level. The third part is about brain imaging, mostly about non-invasive methods. And the last part is a few words about data analysis and about mostly about EEG, which we will use on the next class. Yeah, and I just want to mention that for the next class there is short homework. If you want to do something with these devices, you should download some programs. You can also see it on the presentation. I'll send you a link. So Cortex on Europlay Pro for laptop. So you should bring your laptop on a class. You can work in pairs or in groups, but it's better for everyone to have its own laptop, because sometimes, because not every laptop can connect to these devices. So if you have, for example, 2 laptops for 2 people, it's easier to connect one of them to the neural interface. Or you can also do some measurements with the help of your smartphone with Android. In this case, you also should download Europlay for Android from this site. And maybe later, after the next lessons, it will be, if you want to do something with the data you will take from these neural interfaces, you should download Jupyter iPad notebook and some appropriate environment, and I can also send you an instruction if you want to do something with the data. Is that clear?
Yes.
Yes, thank you.
Okay. So I think you remember this picture. This is a kind of timeline for you for better understanding the chronology. And in the end of 19th century, Golgi staining method was discovered, and it was the very beginning of neuron doctrine. And the first method of non-invasive detection of brain activity, EEG, was developed in 1924. But first method of imaging, such as MRI and later fMRI, they were discovered only in 16th, and almost at the same time as first artificial neural network. One more thing you should know is that there are two types of tools. Two types of tools to work with the brain: methods for diagnostic and detect brain signals. So when you measure something in the brain and you want to know how the brain reacts on different stimuli. And the second type of tools are methods of stimulation of the brain and neurons. And we will mainly talk about signal registration methods. So about these methods and less about these methods. So with the help of second techniques, I mean, with the help of manipulation techniques, you can change the behavior of people. Okay. So first of all, scientists learn how to record and transmit a signal to the brain using electrodes. As I said, at the beginning of the 20th century, the first human EEG was recorded, and Hans Berger did it. As you remember, the patch clamp method was invented in the 70s, and it became possible to record currents through single channels. And electrical brain stimulation was known in 19th century, so before the patch clamp. One more timeline about imaging methods. As you can see here, the date of birth of cognitive neuroscience is just the end of 18th. And that's because the main method of human brain activity visualization, fMRI, was created in 1991. So it means that formally cognitive neuroscience is a rather young field. So its age about 30 years. To get down to a discussion of all the methods for studying the brain, let's present them in one graph. In this graph, the axes are the spatial and temporal resolutions. So you see the recording methods with the highest temporal and spatial resolutions are invasive. So it means that we need to damage brain. Invasive means we should destruct some structures, some neurons in the brain. Another view of the same graph. Here the vertical axis shows which structures are visible using a particular method. So the mesoscopic level is when individual neurons are no longer visible, but individual neural columns or zones are visible. So this looks like that. You can't see individual neurons, but you can see individual zones. And fMRI captures the part of the mesoscopic level. And the third presentation option, here the color indicates the type of method, invasive or non-invasive. The green is non-invasive, and the red one is invasive, and the blue one is natural. Okay, so you see that in fact there is a division into two registration methods that allow you to record the activity of individual neurons and groups of neurons, and imaging methods that allow you to see the activity of brain zones. All methods of registration of neurons are invasive. So you can't see the individual neurons without damaging the brain. At the moment, it's impossible. And let's move on to these methods. Methods for recording the activity of individual neurons. So the first method is a single unit recording. Single unit recording provides a method of measuring the electrophysiological responses of a single neuron using a microelectrode system. When a neuron generates an action potential, the signal propagates down the neuron as a current which flows in and out of the cell through excitable membrane regions in the soma and axon. A microelectrode is inserted into the brain, like here, where it can record the rate of change in voltage with respect to time. So this microelectrode must be fine-typed, low impedance conductors, and they are primarily glass micropipettes, metal microelectrodes made of platinum, iridium, or even iridium oxide. Microelectrodes can be carefully placed close to the cell membrane, allowing the ability to record extracellularly. So on this picture, you can see very famous part of the brain. Do you know this part of the brain?
Almost.
Sorry?
Cortex.
Not a cortex.
The gray matter.
Of course, somewhere here there is a part of gray matter, because gray matter means neurons, neurons bodies. Okay, this is a hippocampus. Usually it looks like that, and mostly the hippocampus of a rat or a mouse. And so the dentate gyrus is the enter of perforant path in the hippocampus from the cortex. So here there is a cortex, and then through mucous fibers, the signal goes through CA3 and then to CA1 and later and later.
So this is invasive or not?
Of course, this is invasive. Yeah. So invasive means we should distract the brain and the scalp, and so the invasive means something we can do only in a lab. And for example, we can do non-invasive with ourselves. So on the next lesson, the next class, we will do some non-invasive methods. Okay, so we won't measure something like that. The single unit recording are widely used in cognitive science, where it permits the analysis of human cognition or monkey cognition and cortical mapping. Here you can see an illustration from Foster's paper from 1997. It's a kind of traditional paper. He researched working memory. So the working memory of the brain is like the working memory of a computer. This is what you remember at the current moment when you work with material. For example, if you want to add some numbers, you need to use working memory to remember these numbers. And in this case, the monkey was given a task. One of the circles that it had to press was highlighted. And after a period of time, after 15 seconds, monkey pressed the corresponding button. So all this delayed time of 15 seconds, the monkey kept the desired circle in working memory. And here we can see the spikes from the neurons from prefrontal cortex. So we can see that the activity are much higher during the working memory period, during this delay. So this is one of the methods how researchers can understand which neurons are responsible for what. The patch clamp technique is a laboratory technique in electrophysiology, and it's used to study ionic currents in individual isolated living cells and so on. So patch clamping can be performed using the voltage clamp technique, and in this case, the voltage across the cell membrane is controlled by experimenter, and the resulting currents are recorded. Also, there is a current clamp technique when current passing across the membrane is controlled by the experimenter, and the resulting changes in voltage are recorded. So the voltage clamp and current clamp. It's named by what you control. During a patch.
So can I ask a question? What's the difference between the patch clamp and the single unit recording, something like this?
When you use a single unit recording, you use just electrodes, not micro pipettes. So you measure the voltage near the cell. It's extracellular recording. So very near, not on the membrane of the cell. Okay.
So patch clamp is more accurate since it gets inside the cell.
Yeah, it's more accurate. So with the single unit recording, you just see the voltage near the cell. So you can register different action potentials, but you can't register the potential on the membrane. So if you haven't got potential, you won't see anything. But in the case of patch clamp, you can see very tuned things, even currents through single ion channel, through one molecule.
So all these techniques just try to detect the workload happening inside the brain. I mean, just like how much active is the brain at current moment, and we just try to interpret that by other means. I mean, this is the output of all the techniques.
Yeah, but
like in the last figure of the monkey, he was trying to differentiate between blue and red, but the technique only gives us the result of the workload. Like at this specific segment, it was too much like working compared to the first segment, something like this. But it doesn't tell us like whether he was thinking of this color or even thinking of a different topic. You get what I'm saying?
Yeah, of course, we can measure just the activity of the neurons with this method. So this part of lecture is about measuring the activity of certain neurons.
Yeah, I'm just asking about all types of works on neurons or brain, just trying to detect the activity. Like this is more active now than like 2 seconds ago, something like this, using the potential difference.
That's right.
Okay.
And also patch clamp, frankly speaking, patch clamp it is not used in the brain, you see. It's used in vitro. So here there are some neurons which are still can function, but they are not in the brain. They are not in a living brain. So we usually use patch clamp if we want to register some particular neurons. And during a patch clamp recording, a hollow glass tube known as a micropipette or patch clamp pipette filled with an electrolyte solution and a recording electrode connected to an amplifier is brought into contact with the membrane of a nasolytic cell, and another electrode is placed in a bath surrounding the cell or tissue as a reference ground electrode, and electrical circuit can be formed between the recording and reference electrode with the cell of interest in between. So it's how it looks like in real life. It's microscope and patch clamp electrode. The next method of registration is a microelectrode array. A microelectrode is an electrode used in electrophysiology, either for recording neural signals or for the electrical stimulation of neural tissue. Microelectrode arrays, or MIR, are devices that contain multiple tens to thousands microelectrodes through which neural signals are obtained or delivered, essentially serving as a neural interface that connects neurons to electrical circuitry. There are two general classes of mirrors. Implantable mirrors are used in vivo. Here they are. And non-implantable mirrors used in vitro. So on this slide, you see microelectrode arrays for registering in vitro neurons. That is that there are neurons in a culture in a petri dish, and we register their activity. So it's not, you can see it's not to tune. So there are quite a lot of neurons around this electrode, and that's what we see as the activity of this electrode from the surrounding neurons. And okay, so this slide shows a mirror implanted in vivo. So with the help of this microelectrode arrays, you can remove a signal from the cerebral cortex, and a person can also control the neural interface using brain activity. The length of this electrode is not great, so they do not penetrate into deep skull structures of the brain, only into cortex. So you can see there is such a microelectrode array, and it's rather small, and it's how it looks like in the brain. So here in the center we have got motor cortex, and that's why such electrodes, they are transplanted into the central part of the brain, and with the help of neural interfaces, people who can't do something with the help of their arms, for example, whose connection of arms and the brain is damaged, they can do something with the help of robotic arm, which is So we can do it just from the cortex, from the brain. Okay, and now there is a question for you. Do you know where the picture on the left comes from?
What do you mean?
Have you ever seen this?
Elon Musk.
Yes, of course. That's an illustration of Neuralink of Elon Musk. So, in fact, Neuralink is the same microelectrode array, but the electrodes in it are much longer. And due to this fact, such an implant can allow receiving signals and stimulating deeper areas of the brain, not as the usual microelectrode array. And there is another difference between Neuralink electrodes and conventional ones. It's that Neuralink electrodes are softer, and therefore the electrode make its way between neurons, and they do not damage the neurons. Okay, and now let's move on to the next method. Here on the picture, there are methods.
I have a question, please.
Okay.
So
microelectrodes array.
Yeah, it is more accurate than battery clamp.
No, microelectrodes array, it's like almost like many single units array, but not exactly. So you can't measure the membrane potential on the membrane with the help of microelectrode arrays. You still measure the potential somewhere near the neurons. But if you have got, for example, 3 electrodes, not only one, and you can calculate signals, for example, from 3 neurons. So if one electrode has signals from many neurons, you can distinguish them. You can find if this spike is from neuron number 1 or from neuron number 2. So, but if you have several electrodes, you can write some equations and calculate which spike is from which neuron. So it's a little bit more accurate than single unit recording, but it's less accurate than patch clamp.
Okay.
Okay. So here there is a Golgi staining technique. We've already discussed it before, and since these years we have more interesting techniques. So have you ever seen this picture or something similar?
Yes.
So do you know what is it?
What I saw, it was a different staining for transcriptome, like the transcriptome of some cell, like with different green fluorescent proteins, something like that. Got nothing to do with neurons. Maybe it's close. Yeah,
it's quite close. So this method is called brainbow, and it is a process by which individual neurons in the brain can be distinguished from neighboring neurons using fluorescent proteins. So by randomly expressing different ratios of red, green, and blue derivatives of green fluorescent protein in individual neurons, it's possible to flag each neuron with a distinctive color. So While earlier labeling techniques allowed for the mapping of only a few neurons, this new method allows map more than 100 differently mapped neurons to the simultaneously and differentiality illuminated in this matter. So in some way, brain-board is a kind of optogenetics. Optogenetics most commonly referred to a biological technique that involves the use of light to control neurons that have been genetically modified to express light-sensitive ion channels. As such, optogenetics is a neuromodulation method that uses a combination of techniques from optics and genetics to control the activities of individual neurons in a living tissue, even within freely moving animals. So in some usages, optogenetics also refer to optical monitoring on neuronal activity and control of biochemical pathways in non-neuronal cells. Also, these research activities preceded the use of light-sensitive ion channels in neurons. Neuronal control is achieved using optogenetic activators like channel rhodopsin. Do you know what is rhodopsin? Channel rhodopsin?
Transmembrane protein.
Sorry?
Transmembrane protein.
Yeah, and where we can find this protein?
Eyes.
Yeah, that's right. So the rhodopsin is a kind of protein which can react on photons. So we change its properties when the photon is on this channel. Neuronal control is achieved using such optogenetic activators like channel rhodopsins and halorhodopsins and any other rhodopsins, while optical recording of neuronal activities can be made with the help of optogenetic sensors for calcium, neurotransmitters, or membrane voltage. And control of activity is restricted to genetically defined neurons and performed in a spatio-temporal specific manner by light. Optogenetics allows to modify only a certain group of neurons so that it's sensitive to optical control. Also, it allows to excite this group of neurons. For example, in 2013, the group of authors created fake memory for a mouse thanks for this optical activation of individual neurons in the hippocampus. So they found these neurons which were activated when the mouse was in a kind of labyrinth, and then they activated these neurons again when the mouse was in another place. So, and it tries to go as it was in the first labyrinth.
Can you repeat this again? How someone can maneuver or intervene with the memory using optogenetics?
Oh, okay. We are going to talk later about and more about the hippocampus. But for example, in hippocampus there are special place cells. Have you heard about the place cells in hippocampus? Okay, so when, for example, a mouse or a rat is in a certain place of a labyrinth, usually we can find some special neurons in hippocampus, which we call place cells, which are active when the mouse is in this current place. And when the mouse goes to another place, we have another place cells which are active here.
You say place cell, like you mean location cell, like cells responsible for remembering locations?
Yeah, that's right. So rats and mouses, they have, and maybe people, but we know more about mouses and rats. So they have some special neurons in hippocampus which are responsible for certain places. And we will talk later about people. So people have special neurons which are responsible for different concepts. For example, a neuron of Jennifer Aniston, or maybe a neuron of, I don't know, some special building of MIT campus, or so on. Okay, and so we can detect these neurons which were active when the mouse went through the labyrinth, and then and if the mouse is in the first time in this labyrinth, some special genes are active in the neurons which are active at the first time here. So we can connect our optogenetic devices with the neurons which produce these genes for new memory. So we can find the path, some kind of path of these new memories in the mouse brains, and then we will activate with the help of light the same neurons which were active at the first time when the mouse was in the labyrinth. So they will be active when the mouse is not in a labyrinth. For example, imagine that I talk with you and I'm in this lecture, and maybe this is my first experience on this lecture, and someone can find the neurons which are active during this my experience, and then I go away, for example, in my bathroom or in my kitchen, and someone activates the neurons which were active here on the lecture, and in my kitchen I think that I am in this lecture, so because these neurons are active. Okay, so frankly speaking, it's not so easy, but you should just understand the concept that it's possible.
Because this actually is like a science fiction, you know, the movie Inception, when you plant an idea inside someone. So at that time it was like not real, but reading this.
That's what scientists already can do, but the problem is that we do it only with rats and mouses, because no one allows us to do it with people.
I can volunteer. Okay.
Okay. And optogenetic methods make it possible not only to control the excitation of neurons, but also to read their activity. For example, as you remember, when a neuron is fired, there is an increase in intramembrane calcium. If there are special proteins in a neuron that glow when combined with calcium. You see neurons that have fired. So this is called calcium imaging. For example, here and here, kind of calcium imaging. We discussed methods that are indicated here in pink and in red, and let's move on to green. So next part is brain imaging. It's almost the entire classification of brain imaging methods. So there are three major groups: techniques involving radiation, techniques involving magnetic fields, and techniques involving electrical activity. Some of them look like this. That's not a meme. That's real pictures of different brain imaging methods. So let's start with computed tomography. It was very popular during the COVID-19, as many had lung computer tomography scans, but for the brain, this method is also used. So a CT scan or computed tomography scan, formerly known as a computed axial tomography, is a medical imaging technique that is used computer processed combinations of multiple X-rays measurements taken from different angles to produce tomographic cross-sectional images or virtual slices of a body, allowing to user to see inside the body without cutting. So the personnel that perform CT scans are called radiographers or radiologic technologicals. So it's kind of combined X-rays. These pictures shows two more methods. So do you know what are these methods? What's on the left? Do you know it?
Not really.
Not really. Okay, and on the right?
You mean the name of the technique that was used?
Yeah, do you know the name of this technique?
Exposure to magnetic field or something like that, or radiation.
And these are fMRI on the left and PET or positron emission tomography on the right. It's how they can usually look like. So positron emission topography or PET is a functional imaging technique that uses radioactive substances known as radiotracers to visualize and measure changes in metabolic processes and in other physiological activities, including blood flow, regional chemical composition, and absorption. So different traces are used for various imaging purposes depending on the target processes within the body. PET scanners can incorporate CT scanners. PET scan images can be reconstructed using a CT scan performing using one scanner during the same session. PET is both a medical and research tool used in preclinical and clinical settings. So it's used when we want to know if there are any tumors, for example. So they are used in oncology and for the clinical diagnosis of certain diffuse brain diseases, such as those causing various types of dementias, maybe like Alzheimer's disease. magnetic resonance imaging, MRI, is a medical imaging technique used in radiology to form pictures of the anatomy and the physiological processes of the body. MRI scanners use strong magnetic fields, magnetic field gradients, and radio waves to generate images of the organs in the body. And MRI doesn't involve X-rays or the use of ionizing radiation, which distinguishes it from CT and from PET scans.
So CT and PET use ionizing radiation?
CT uses X-rays.
Positron emission use what?
X-ray, it's radiation.
I know X-ray. I mean, like what type? You said ionizing radiation, and those are listed in the green and non-invasive techniques, right?
PET uses ionizing radiation.
I mean, ionizing radiation is already invasive. I mean, it damages the cell. You know, I mean, so like it's not totally safe, or is it?
Yeah, it's not totally safe.
Okay, so it's invasive.
Yeah, I think it's determined on the concentration of the radiation.
Yeah, that's right. But you shouldn't do PET scans or X-rays very often. So I think you usually do, for example, X-rays once a year or not very often. And so the same is about PET scans. But you can do MRI and fMRI quite often. So this is the difference. It's much more safer. And it's used in research more often, maybe also because it's safer and because it's cheaper. So in most medical applications, hydrogen nuclei which consist solely of a proton that in tissues create a signal that is processed to form an image of the body in terms of the density of those nuclei in a specific region. So given that the protons are affected by fields from other atoms to which they're bonded, it's possible to separate responses from hydrogen in specific compounds. To perform a study, the person is positioned within an MRI scanner that forms a strong magnetic field around the area to be imaged. First, energy from an oscillating magnetic field is temporarily applied to the patient at the appropriate resonance frequency. Scanning with X and Y grading coils caused a selected region of the patient of the experience the exact magnetic field required for the energy to be absorbed, and the excited atoms emit a radio frequency signal which is measured by a receiving coil. And fMRI provides a measure of hemodynamic, so it's blood-based activity in the brain, and is based on the premise that neuronal activation increases oxygen demand of neurons and related cells, leading to additional blood flow carrying oxygen molecules to the region. This can be measured using BOLT, or blood oxygen level dependent activity. So MRI and fMRI, they are indirect methods, so we don't measure the neuronal activity with the help of fMRI. We just measure blood oxygen level dependent activity. So you see the difference? So we know that there is a connection between neuron activity and blood oxygen level, and they should correlate, but we don't measure the activity of neurons, we measure the activity of blood oxygen level. Both PET and fMRI rely on a subtraction method where brain scans for different experimental contrasts are subtracted from each other. A frequent condition for...
Okay, can you please come back to the last slide and say again what it was about?
This slide?
Yeah. What's happening?
In fMRI, for example, we measure the changes in between different bold signals. For example, some control activity at the moment, yes, and later when there is some activity in the brain. And to find the signal at this moment, we should take this signal.
And this control is for the same brain, or it's just like a already known control?
It's for the same brain.
And how to know the control from the stimulation? Like you stimulate the brain, then you detect it, then you stop the stimulation and detect it, and you minus this from that, and you get the difference activity.
In fact, we use a kind of MRI signal, and then to find the fMRI signal, we do fMRI minus MRI.
But how do you subtract one technique that depends on something and from another? Like you said that fMRI blood-based, it doesn't scan the neurons, just say the level of oxygen. While the other one is different. Like how do you, or is it like this? Okay, I don't know.
Yeah, but MRI is not blood-based. So we use MRI when we want to see the structure of the brain. So we see white and gray matters with the help of MRI. But in fact, when we use MRI, we don't see this flow of blood oxygen. No, we just see almost the pictures. They are almost they looks like MRI. And to find this blood oxygen level, which we just should do minus MRI activity.
Okay.
So, but very often we measure the brain in a state of rest, so without any tasks. But now we know that while activity during a resting state may not be directly related to the tasks being performed in other states, we know that humans, they are constantly thinking or maybe imagining or feelings, anticipating something or remembering, because there is always some kind of activity in the brain, in a living brain. So we can't stop the blood oxygen activity. PET was developed before fMRI and provides a measure of metabolic activity in the brain, and it's very expensive to use, and it's not as safe as fMRI, because we use a radioactive tracer here. However, it's expensive, invasive, and slow, and a more popular method currently is MRI and fMRI, and based on some different principles. So to sum up, these methods we mentioned, they can be divided into two categories, direct and indirect. So direct brain signals are usually electromagnetic. They include EEG, MEG, single cell electrical recording. Here we measure some electroactivity of neurons, or maybe summed up electrical activity. And for indirect methods, we measure something else. For example, we measure blood flow or brain metabolism. So fMRI, MRI, and PET, they are indirect methods. And of course, all brain methods have their pros and cons. So now try to guess these two methods. So what's on the left? Do you know it?
It's like microelectrode.
No, it's non-invasive. Okay, I guess you don't know what's on the right either.
Try to name us few and we'll back from them. Like, tell us, is it this or this or this? And we will say, no, we don't know.
Okay, no, I'll tell you the answer. Sorry, just on the left, it's EEG. So EEG is electroencephalography. And on the right, it's MEG, so magnetic encephalography. And let's talk about them more. So electroencephalography is an electrophysiological monitoring method to record electrical activity of the brain. And it's typically non-invasive, with the electrodes placed along the scalp, so on your head. Also, sometimes it can be invasive, but it's called electrocorticography. And EEG measures voltage fluctuations resulting from ionic currents within the neurons of the brain. So we measure the potentials on the surface of the brain with the help of EEG. Now, neurointerfaces are widely used. So the main principle here is also EEG. And the neurointerfaces are more transportable, sometimes with fewer electrodes and often with dry electrodes. So there are two types of electrodes. There are ordinary wet electrodes, like here on the left. They use a conductive gel, and dry electrodes here on the right. They contain flexible legs made of conductive plastic. And on the next class, we will have neurointerfaces with dry electrodes. Magnetoencephalography is a functional neuroimaging technique for mapping brain activity by recording magnetic fields produced by electrical currents occurring naturally in the brain using very sensitive magnetometers, arrays of squids or superconducting quantum inference devices. There is some medical use for.
Can I ask a question about the last two? Last two classified into the direct imaging, right? Not indirect.
They are direct. That's right. Yeah.
And they use magnetic techniques. So what's the difference between this and MRI?
What's the difference between EEG and MEG? Yeah.
No, both and the one that use magnetic resonance to MRI?
MRI, fMRI measures the blood flow, in fact.
That's fMRI, but MRI without the F.
MRI measures just the structure of the brain. It's not functional method. See, maybe I find some. So maybe, well, this is MRI. So there is no any activity here. So it's it's almost like CT. But fMRI is a functional MRI, and here we see some kind of activity. So for example, here there is fMRI, and the colored regions, it's the difference between the current activity and between the MRI. So the structure. That's what I thought about. Okay.
So the last two can be used for like clinical use to diagnose something like tumors or whatever. Those two, yeah.
Yeah, or all of them can be used for some kind of diagnosis, but in clinical, these two direct methods, they are usually they are used for epilepsy and some seizure disorders. So they usually used for some unusual activity, for some unusual brain activity. So if people, if doctors, if they want to find if there is some tumor or not, they should use MRI and PET because it's got better better space resolution. And if we want to find some seizures, some epilepsy, something in the activity, in the electrical activity, it's better to use EEG or MEG. Okay, or maybe sleep disorders, because it's also connected with the activity so much. So EEG measures the electrical component, and MEG measures magnetic component of the field. Since the current mainly flows in the cortex orthogonal to the surface of the cortex, the EEG is most sensitive for measuring currents in the sulcus, and MEG is more sensitive for measuring the current in a gyrus. Sulcus and gyrus. You can often see topographic images of the brain activity made with EEG and MEG, but EEG and MEG are not imaging methods, so these pictures are not quite accurate. There are two types of tasks connected with EEG. So the direct and inverse problem of EEG or MEG. And the direct task is known that what sources of current are inside the brain to find the distribution of potentials on the surface of the skull. And the inverse problem is knowing the potentials on the surface, find sources inside. And the second problem is not solved, but there is some software for the probabilistic solution of the inverse problem of finding sources in the brain by EEG or MEG. So we can measure the potentials on the scalp and then reconstruct the currents in the brain, but it's a kind of probabilistic solution, so not very tuned. Okay, and some words about EEG. So the most common and oldest of the methods for analyzing in EEG is spectral power methods or rhythms analysis. We will talk a little bit on the next lecture about alpha, delta, beta, theta, and so on rhythms, and what do they mean. So if we want to find out the spectral power or the power of the rhythms, we can calculate them with some techniques. The second thing you can do with EEG, and also we can do it with fMRI too, is functional connectivity analysis, and it looks something like here. If you want, we can also calculate some functional connectivity with you. There are three types of connectivity: structural, functional, and efficient connectivity. The difference is that structural connectivity analysis analyzes the presence of connections between two regions of the brain. Functional connectivity indicates whether two parts of the brain are active together or not. For example, there may be rails between two cities, but this doesn't mean that trains run between them. And effective connectivity also tells us which way the trains or the activity are going. Another method of analysis is the search of sources. In fact, this is the solution of inverse problem when we are trying to draw a picture similar to fMRI with the help of EEG data.
What is the forward and inverse problem again?
Sorry, what
are the inverse problem and the forward one?
Okay, when we measure EEG, we know just some potentials in some points on the surface of the head. Okay, and we don't know the activity in the brain. So source means the source of the potentials on the surface. So there are some currents in the brain, and if we want to find these currents, it means we solve inverse problem of EEG. But if in some way we know the activity inside the brain, the currents in the brain, and we want to know the potentials on the surface, that's called forward problem.
So direct and indirect.
Yeah, that's the same. And the last way is machine learning. So we can simply fix some individual states. For example, we can measure some EEG when I open, when my eyes are open, then I close eyes, and we measure some EEG again. Then we train classifiers or neural networks to find a given states among our measurement. So I can open and close eyes again and again, And with the help of some classifiers on neural networks, we can find where is the open eyes and closed eyes and so on. But this is a quite simple problem, and there can be more difficult problems. For example, we can detect states when I'm trying to solve some difficult problem and when I do nothing. Or we can try to find state when I'm concentrating on some ideas and when I'm not focusing on something. Okay, so and we use some additional methods, for example, such as independent component analysis. In some way, it's part of machine learning methods, but it's usually it can be usually used for finding some artifacts. So here, for example, we can see some mixed signals, and with the help of independent component analysis, we can find out some different sources from here. And when we use it for finding artifacts, it looks like that. For example, here we can see a very noise component, so it's rubbish. Here we can see artifacts kind of blinking, so when my eyes closed and opened, and we should throw it out too. And then we mix the other signals, so the true signals, and after that we'll have a more clear signal, for example, for EEG. And when we use independent component analysis, for example, for fMRI, it looks something like that. So if one of the components is looks like many dots, it's kind of noise. And if it looks like rather big parts of a brain, that's a normal signal. And so we should find out different components and understand which one is noisy component and which one is okay. And then we sum up some good components, and so we have clear signal. So I think that's all for today. On the next lesson after, not on the next week, but after a week, we'll have a class in MIPT, and we'll have a small lecture about attention and meditation, and we'll try to measure something with the help of our devices, neural interfaces, but you should bring your laptops and to install some programs. And don't forget about the test after, I think after 2 hours, because I'll have a lecture right now. So thank you.